Please be seated. Esteemed PhD candidate, the College of Deans of this university, represented by us, has read your PhD thesis and the propositions that go with it. And we are ready to hear your defense then. Please be seated. Does anyone in the audience wish to exchange ideas with the PhD candidate? I see that this is not the case, so it's then my great pleasure to invite for the first position Professor Jimenez, who came to us from the University of Barcelona. Professor Jimenez, the floor is yours.
Thank you very much, Director. I first of all want to acknowledge and to thanks to the candidate and the mentors for his excellent work and for giving me the opportunity to be here, because I'm really happy that you did the work, because this is the reason why which I am here. So this is the first question. The second question is that I'm very interested in your work, because I have been working for a long time trying to develop some kind of alternative system to the studies in the middle of animals and humans. For this reason, I acknowledge the extraordinary insight that you have placed here. So thanks again. And the first question came to me because it was almost immediate when I started to look at your work, and this was the title. I get very surprised, the 5 millimeters closer to human studies. And then I said, well, this should be like a poet or something like that, because so I'm interested in to know how this title came to you.
Hi, learned opponent. Thank you for your kind words and compliments, and thank you for coming here today. The title indeed, it was a inspiration. We came up with 5 millimeters closer to human studies because of work with slices, especially human slices, and 5 millimeters in this case, they refer to the size of slices that we make. They are precisely 5 millimeters in diameter. So by using human slices, which are this small, we hope to contribute to advance the field of fibrosis closer to human studies, so we have more successful human studies completed with antifibrotic treatments becoming available by using this model. Hence, 5 millimeters closer to human studies.
Thanks. I should say that sometimes scientific literature is very tough to read, and this is not the case with your thesis. Thank you. And the second question is more, how to say, philosophical, in terms that it's tough to believe that the system can be useful for, as in an extrapolation, any kind of tissue or organ and any kind of compound that you want to check. In particular, because the structure of the organs is very different, and some of them are much more consistent than others. The type of cells that you have there are different. And on the other hand, the type of drugs that we want many times to check have very different characteristics in terms of even delivery to the cells, be accessible to the different type of cell, etc. So, could you justify so optimistic concept in terms of your model?
If I understand correctly, you're referring to if we can test the whole range of different compounds with different mechanisms of action and delivery to cells within our system. And I think the answer would be yes, because it's quite a versatile model. So in my case, in this thesis, yes, we did a simple addition of compound to the culture media, but you can also try to use nanoparticles loaded with the compound. You can do targeted constructs delivered to slices. I think there's also some work with liposomes, exosomes, which could be tested within our system. So even if the drug doesn't have a direct, let's say, uptake mechanisms by the cells, you can always try to overcome this barrier and use one of those targeted delivery systems and have an effect.
Okay. And so the last question is that we have always saying that some findings that we have with animals, in particular with rodents, rats and mice, when we want to translate to humans, are missed. And we always said the rats are not the same than humans. So in this case, how you want to overcome or pretend to overcome this problem, because the findings that you show here mainly are in rodents.
I am myself a big advocate for using human tissue, even for making slices. I would still, first of all, use human tissue. I'm not debating the additional value of using murine tissue from mice and rats, because they do provide an excellent platform for also preliminal screening for toxicity, for drug metabolism. They are different from humans, but as my colleague was mentioning before, there is still an overlap between how the mouse responses in mice and responses in human. And if you know what the overlap is, and your research question falls into that margin, you can be confident that what you're researching in mouse or rat will be reflected for what happens in the human, for example.
Okay, thank you very much.
Thank you very much, and once again, thank you for traveling all the way from Barcelona, Professor Jimenez. It's been a great pleasure to welcome you here. For the next opposition, I'd like to call upon Professor Faber from the Department of Gastrointestinal and Liver Diseases. Professor Faber.
Thank you, Mr. Rector. Esteemed PhD candidate, mevrouw de promovendus. Of course, I also would like to congratulate you first with a beautiful thesis that I read with really great interest, and also would like to congratulate your supervising team with this very nice research. And again, I'm honored that we can, let's say, that I was allowed to, read this already in an unpublished form. So it's really exciting work. So I'm a bit sad about the first opponent, because he took away my first question. And that is about the 5 millimeter closer to human studies. But I have a little addition, and again, I need one of the parliament to read one of your propositions, and that is actually proposition 8. So,
one who works with intestinal slices goes through denial, anger, bargaining, depression, and flapping acceptance.
All right, thank you very much. So the question is, did you really get even 5 millimeters closer to human studies using intestinal slicing?
Hi, opponents. Thank you very much for your compliments and your kind words. To finish the 5 millimeter discussion, so yes, intestinal slices are, in my experience, are more tricky than slices that we make from other organs. And I think I am at the acceptance stage right now. So,
but what do you accept? Do you accept that they don't work?
Yeah, I accept that they are different, and it's harder to work with them in the sense that the responses that we see in intestinal slices are very different from what we see in liver and kidney. And in terms of efficacy of the drugs that we test in intestinal slices, it's also a bit frustrating because the compound, the galvanistic compound, the compound that works everywhere, basically doesn't work in intestinal slices either. And we had additional problem with intestinal slices from humans because the study population, the donor samples that we get are quite different from each other. So, and we didn't find the way to overcome this big variation. So we tried to stratify patients based on, for example, the patient samples based on collagen content, but it didn't work that well. So I think before we can continue our research with intestinal slices with human, we have to first somehow deal with this high inter-sample variation.
Because I was triggered by indeed the human studies where you take samples from stenotic tissue from Crohn's disease patients and flanking, let's say, kind of healthy tissue. And if you compare this with, let's say, liver research and kidney research, I mean, the remarkable thing is that you actually do not see a difference between the stenotic tissue and the normal tissue. So how can that be?
Yeah, that is indeed what we also saw in chapter 4. I think it's chapter 4.
Yep.
So yes, when we had ileum from healthy patients before culture, and we had ileum from Crohn's disease patients also before culture, normally you would expect a big difference, right? So between healthy and disease state, but that was not the case. The problem.
So they are not really patients?
No, they are. They are patients. The thing is that when we prepare intestinal slices, we actually culture only the mucosa. So we strip all the way all the other layers from intestine, and what we culture is mucosa. But in Crohn's disease, it's known that inflammation is actually transmural. So all the layers can be involved. Layers of GI tract can be involved in the process of inflammation and fibrosis. So maybe when we culture only mucosa, we lose that part, that essential part from other cell layers, and that's why we don't see the difference.
So then you are claiming that in let's say, stenotic fragments in the intestine of a Crohn's disease patient, there is actually healthy mucosa covering this. Wouldn't that be spectacular?
Yes, spectacular. I wouldn't still call it healthy because also healthy healthy tissue that we call healthy is not like absolutely healthy because they still come from patients who have disease or injury, so they have some level of background. This mucosa, yes, appears to be not, from Crohn's patients, appears to be not as diseased as we would desire it to be, but I wouldn't call it completely healthy, so those patients are not that lucky.
So together with your promoter, I actually are working now on a project proposal for a big company to actually work with intestinal slices. So I'm a little bit lost now. So how should I convince them that these intestinal slices are still useful to do this?
They are useful, but you have to really make sure that they are aware that there are certain technical problems they face, and there are some steps of
Companies
optimization.
normally do not realize that there are technical problems, and they don't just want to have an answer to their specific question.
As soon as they overcome some of the obstacles we face now, it's good to go.
So we should really have a development stage also in such a project, I guess.
Yes, if we follow up on chapter 7, so we extend the culture period, and I think our preliminary data with human intestinal slices is quite promising. at least that is will be already one step forward to actually implement this system into drug development, because then we have long-term incubation of slices with all the cell types living and functioning.
Thank you very much. I'm checking the record, but I'm not sure.
You can have one very short question, and I would ask candidates to answer also shortly.
Well, thank you very much. This links a little bit to what you just said. So we were trying to optimize the culture conditions, and focusing, I think, on stem cell, let's say, compounds that promote stem cell proliferation. But especially for the intestinal ones, would it also be useful actually to look at the microbiome and metabolites that come from the microbiome that actually would support the growth of these epithelial cells? Because I think that is specifically missing in the intestinal slices for intestine.
Definitely, this is a very interesting area of research, like trying to combine microbiota and tissue, intestinal tissue, to see what is the interplay between the two, how one affects another, and how we can balance it.
What would be the prime compound that you would like to test?
Prime compound, maybe one of the ones that are already in clinical trials, and see if they give some promising results also in our system.
Well, I mean, let's say coming from the microbiome to support the growth of the intestinal epithelium.
I mean, from the microbiome, it doesn't come to my mind.
I would start testing short-chain fatty acids, because that is, let's say, a real specific product from the microbiome to support the epithelium. I think my time is over for this position, so thank you very much for the discussion. I can do it back to the record.
Thank you very much, Professor Faber. I then call upon Professor Hillebrands from the Department of Pathology at the University Medical Center here in Groningen. Professor Hillebrands.
Thank you, Mr. Rector. Esteemed candidate, first of all, congratulations with this beautiful thesis, especially the cover, not referring to the title, but actually to the nice flowers, I think reflecting the 3 different organs with all the different pathways that you analyze. So very nice. I would like to discuss primarily chapters 2 and maybe a little bit about chapter 3 with you. And in that chapter you cultured mouse slices, different organs, 3 different organs, and already under basal conditions at T0 identified quite some differences. My question to you is, what is the explanation, or your explanation, for these differences? For example, with respect to SMA expression or fibronectin expression shown in figure 4.
Hi there, opponents. Thank you for your kind words, and thank you for compliment on the cover. Actually, the flowers represent indeed the slices, and because they come from the same flower, it means they can make different slices from the same animal, and that's indeed what we did in chapter 2. Regarding your question, yes, there were substantial differences in expression of ECM-related genes at 0 hours, so before we culture slices from different organs. And in my opinion, I think these differences come from these organs have different cellular composition, so the ratio of different cell types is different. And that is why, I would expect to see these differences, and indeed we see them. And we speculate that these differences further affect what we see during culture and how the slices, organs, and tissues respond to the treatment.
Yes, so I agree with you that cellular composition is different. And most likely there are also cells involved which highly express SMA, for example, smooth muscle tissue, which is highly abundant in the intestinal tissue, but not that much in liver or kidney. But also those cells might be not extremely involved in fibrosis. So my question to you is, does it make sense, and is it a proper comparison to compare these organs with each other from the point of view in their response to fibrotic signals?
I think it's still a valid comparison, just because we don't limit ourselves to comparing them only before culture, because, as I said, we expect the differences there, but we wanted to see how these differences evolve during culture. And because culture is such a uniform, imposed condition, that you would want to see how the slices from different organs behave during that injury, stress, under stress, and how they respond to compounded targets, core mechanism of fibrosis, TGF-beta pathway. Because we know, even if there are different cell composition organs, the TGF-beta pathway is involved in fibrosis in all of these three organs. And then I think Venus has showed that the TGF-beta pathway is involved in indeed all organs, but it's involved to a different degree.
Okay, thank you. I would like to move on to actually the same figure, figure 4 in chapter 2, in which you show data obtained using messenger RNA expression analysis, but also using protein analysis. And actually I got a bit worried when I compared the results, and maybe you can bring some relief. And I'm referring to the expression, for example, in SMA, shown in figure 4a. In the liver slices, you do see after culture large reduction in SMA messenger RNA transcript expression, whereas you do see an increase in protein level. In the liver slices, you don't see any difference in messenger RNA expression, but you really do see a huge increase in protein expression. So the question is actually, since a lot of your conclusions are based on messenger RNA expression data, what is your opinion about the interpretation of the messenger RNA expression data? And would it maybe be better also to use another approach rather than primarily messenger RNA expression?
Indeed. Gene expression data, you have to be aware that relying solely on gene expression data might not be 100% indicative of what is really happening. That's why it's always nice to support gene expression data by protein expression data, because a lot of things happen between gene expression and protein expression. Regarding alpha-SMA, it is, in our hands, has the most discrepancies between gene expression and protein expression. And that's why I consider alpha-SMA is not a very good marker of fibrosis in this sense, in this context of slices. Because indeed,
but this is not related to fibrosis. This is whether or not it's related to fibrosis. You do see increased expression in messenger RNA transcripts, or reduction and an increase of the same protein in the same gene in the protein level. It doesn't really tell you something about the relevance for fibrosis.
Yes, but you're focusing solely on alpha-SMA. For example, if you look at HSP47, gene expression is correlating with protein expression. So I do think it depends on which gene protein you look at. You have to know the pattern, how it behaves. So, yeah. Okay,
so you think this is rather the exception than the rule, and you still rely on.
I would call it, yes, alpha-SMA. I would call it more an exception, because also in chapter 4, for example, we measured cytokine release from these slices, and we compared it to the gene expression data derived from NGS, and we saw that it actually correlates the gene expression with protein expression. So I think to some extent gene expression data is definitely indicative of what's happening in protein level, but you have to be sure and you have to do the protein analysis anyway.
Okay, thank you for your answers. Get the work back to you.
Thank you, Professor Hillebrand. It's then my pleasure to invite for the next opposition Professor Bank, also from the University Medical Center here in Groningen, from the Department of Medical Biology. Professor Bank.
Thank you, Mr. Rector. Esteemed candidates, first of all, congratulations with this masterpiece, and also congratulations, let us say, to your supervising team And why I consider it, I consider this as a masterpiece because there's such a huge amount of information stored in it. And I think it can also be used, let us say, for the next generation of scientists. I have been educated this afternoon that this is a thesis or a manuscript, not a book. But for me, through the coming years, it will be a Bible. And as you know, a Bible has been published in 2 volumes, and the first volume we have actually seen about an hour ago. So great work, definitely. I have a question with respect to chapter 2. We can go to page 45. Then you see a Venn diagram over there, so this is figure F. And there you see, let us say, in the overlapping region of the 3 tissues, there are 9 genes that are commonly differently regulated compared, let us say, to the 0 hours, and this is the 48 hours, and this is without a drug. So these 9 genes you have actually mentioned over here at the right side, and collagen 1 is one of them. Can I conclude now from this Venn diagram that in all tissues collagen type 1 is upregulated? Or do I have to take a look to the heat map?
Highly learned comments. Thank you for your kind words, and thank you for being a part of my reading committee. Regarding your question, yes, the answer will be actually we have to look for additional data that is presented in figure, in the same figure but heat map data, or you can go to supplementary material to see what is the direction of change.
Okay,
This,
Now that's actually what I did.
Yeah.
So if you take a look at the same page and then the heat map that is above the F, then you see in the helium that the collagen type 1 is actually downregulated and the other ones is upregulated. So indeed I have to take a look at the heat maps. But what I do not understand from the helium, so from the first graph, is that all collagens are downregulated and all the MMPs, so all the enzymes that are able to degrade collagens, are upregulated. So for me, that is not a fibrotic response. It's the contrary.
Well, yes, you came across the great mystery that we are facing with intestinal slices. I think just downregulation of collagens, just because there is no downregulation of collagens, you cannot say there is no fibrogenic response, because fibrogenic response is an orchestra of many things happening in cells and tissues. And then again, we come just recent question that indeed in gene expression collagen is downregulated, but in this paper, for example, we didn't look actually what's happening with collagen on protein level. So maybe on protein level, there is no such decrease. And for example, in kidney slices, don't mind kidney slices. But yeah, we would need to check what is happening exactly on the protein level.
Okay, so that's more or less similar to the answer from the previous opponent, Professor Hilroth.
Yeah.
Okay, so let's continue. Let's take you to chapter 3 on page 79. You are actually mentioning that, let us say, GP2 was one of the 10 top genes that were downregulated, in this case in human skin and ileum and in the colon slices. So this is one of the top 10 genes that were downregulated. So if you have a drug that upregulates this particular gene, can you use that drug also, let us say, can you test this drug also in an animal model, or do you have to take a look only in human slices?
If you test a drug against GP2, which is commonly downregulated in human slices, I would prefer to test it also in a human setting, so in a human test system. But if you're going for mouse tissues or mouse models, you have to be sure that GP2 behaves in a similar way in mouse as it is behaves in human situation.
Yeah, indeed, but indeed in the mouse it does not behave in the same way.
The answer would be no, you cannot test the compound in mouse system to predict effects in human.
Yeah, indeed. Okay, I absolutely agree with that. Then a final and more broad question is, if you want to develop a drug against fibrosis, do you want to develop a drug that is organ specific, or do you want to develop a drug that treats all fibrotic conditions, irrespective of what kind of organ you're looking at?
I would prefer to have organ specific because I think the idea of treating all fibrotic diseases as having sort of a panacea drug is too idealistic, and certainly developing one drug for it wouldn't. I don't see how it will be possible. And I would prefer to have some organ specificity. So to also